am very excited to introduce our next presenter, Mia Sanchez, who worked on this project with our wonderful late colleague, Dan Gold, and also with Dr. Rowe and Dr. Hauser. I'll let her introduce the project. Dr. Rowe will give her cord and sash to wear at graduation, and we'll have time for questions at the end. So thank you so much, Mia.
Hi, everyone. So to start, I am studying a parasite known as gregarines, and they are a part of a phylum known as apicomplexans. Apicomplexans are numerous infectious parasites, and our gregarines in particular infect nearly all invertebrate clades, so they're found in almost any bug that you can think of. The gregarine species that I focus on is Lauricola migrator, and they infect start hissing cockroaches, so that's our host for today. And some other examples of apicomplexan parasites include Toxoplasma, which causes toxoplasmosis, and Plasmodium, which causes malaria. But as you can see here, our gregarine parasites have the closest evolutionary relationship to Cryptosporidium. Cryptosporidium is a vertebrate infecting parasite which causes the deadly diarrheal disease cryptosporidosis. It also has a very high infection rate. And because of gregarines' close evolutionary relationship to Cryptosporidium, we can use gregarines as a biological model to understand Cryptosporidium. And it's important that we do this because currently there's no preventative treatment against Cryptosporidium. Additionally, Cryptosporidium can be found across numerous sources such as food, water, soil, and contaminated surfaces. It's also important that we study gregarines because they are a very understudied species. Currently, there's only one known transcriptome of grouped internalized gregarine parasites. But this group transcriptome fails to highlight the individual mechanisms between the life cycle stages. Additionally, it's unclear where our gregarines lie on the symbiotic spectrum and if they should be even considered parasites at all. Lastly, although gregarines are known to infect nearly all invertebrate clades, their true infection rates are unclear. So gregarine research provides larger implications for how we can view gregarines as a whole. My research focuses on RNA-seq, where we look at all the RNA within the pooled cells. This allows us to know what genes are being expressed at specific cell stages. Now, a heart and a brain cell contain the same DNA. What makes these cells different is the RNA that's being expressed. In a similar way, for our gregarine parasites contain the same DNA throughout their life cycle stages. But what makes these life cycle stages unique is the RNA that's being expressed. Therefore, our goal is to identify and evaluate the RNA transcripts at the trophozoite and syzygy life cycle stages. We aim to do this through three main research questions. The first being, what genes are uniquely up and down regulated at different internal stages? Upregulation essentially means what genes are being highly expressed for that specific cell stage, whereas downregulation means what genes are not being as expressed or lowly expressed for that specific cell stage. Secondly, we want to understand what genes are being shared among the different internal subpopulations. And lastly, what are the implications for these gene mechanisms that are being expressed? A look into the life cycle stage, starting on the left at the gametesis stage. This is the last internal and first external stage for our parasite. Once our gametesis are exposed to the outside environment, they dehiss to release oocyst chains, which are shown using that yellow arrow there. Those oocyst chains are consumed by our cockroaches and interact with the host vial to exustate and open up to release sporozoites. These sporozoites then partially embed themselves into the intestinal epithelial cells of our host and mature into the next stage, the trophozoite stage. For the purpose of our research, we separated it into two different developmental points, the attached trophozoites and the detached trophozoites. Once the detached trophozoites have matured, they then pair up with one another and move on to the reproductive or the syzygy life cycle stage to repeat the process all over again. I focus on three main subpopulations, the attached trophozoites, detached trophozoites, and syzygy parasites, because of their direct relationship with the host and the ease of identifying between the different subpopulations. Now, this research has been conducted over the past 2 years. And in the first year, I was focusing on isolating our parasite and the RNA from them. In order to do that, I had to dissect our host. So I dissected over 70 cockroaches for 1 summer. And I looked through their intestines, separated our subpopulations, and then isolated the total RNA from that. Once I had the RNAs isolated, I then sent those samples to a company called Novogene, which conducted mRNA sequencing via the Luna program. They then sent those sequences back to us, and we had to build the transcriptomes from there. Because gregarines are a highly understudied species, and there's no reference genome for us to use, we had to use a bioinformatic program known as Trinity, so we could build these transcriptomes unguided. Once we had these transcriptomes built, we were able to create generalized gene expression comparisons for them. One of those being differential expression analysis. Differential expression analysis allows us to see the level of variation between the gene expressions for our 3 main comparison groups. You'll see these comparison groups throughout the remainder of this presentation. The first being attached versus syzygy, detached versus syzygy, and lastly, detached versus attached. Here we see the values for upregulated genes. So this will be genes that are highly expressed in attached, but not as expressed in syzygy, and that same pattern will be found in other comparison groups. What we see here is that the most differentially expressed, or the most different genes, are found between our attached versus syzygy group, whereas the least differentially expressed, or the most similar genes, are found between our detached versus syzygy group. Now, when we look at the downregulated genes, we see the same pattern. And the reason why we thought this is because our detached trophozoites kind of act as an intermediary stage in between our attached trophozoites and our syzygy parasites. So as our detached trophozoites are moving into that syzygy stage, it makes sense that they have more similar genes compared to our attached trophozoites, which have vast morphological differences compared to our syzygy parasites. But in order to look at this, we had to conduct transcriptomic identification. In order to do this, we had to select up and down regulated genes for each of our subpopulations. We were then able to take those lists and put them into CryptoDB. It's a worldwide database that contains all known cryptosporidium genes. And once we had this analysis, we were able to conduct gene expression analysis to kind of paint a picture of the relationships between our subpopulations. One of the ways in which we did this is gene ontology. Now, gene ontology is a way to categorize gene expressions into generalized groupings. So here we see tree maps, which have subcategories for each of our gene expressions, labeled in the smaller boxes with the black labels. These are also paired in larger, even more generalized gene expression groupings that have the white background with them. On our left, we have our upregulated genes, and on our right, we have our downregulated genes. So for upregulated genes, these are the genes that are going to be found in our syzygy parasites, more so than our attached trophozoites. And our downregulated genes are going to be genes that are not as expressed in syzygy parasites, but more so expressed in our attached trophozoites. What we found from looking at the upregulated genes is that syzygy parasites in the reproductive stage are transferring genetic material between one another. So we have genes associated with that, such as the monocarbolic acid metabolic process and the regulation of small GTPase mediated signal transduction. When we look at the downregulated genes, so genes more so associated with attached trophozoites, we see genes associated with growth, development, and response to host stimuli. All things we would expect to see from an attached trophozoite who's feeding off the host and interacting with the host in a more direct way. Here we see genes associated with cellular response to stress, response to stimulus, and protein modification by small protein conjunction. From here, we wanted to get a visualization of these gene expressions. So we took these subcategories and put them into a gene ontology pathway figure. This is more of an arbitrary figure that gains a visualization of our tree map in the previous slide. Each of these circles indicates one of those subcategories from the slide before, and the size of these circles indicate the amount of genes present within that gene ontology group. The color indicates the strength of the significance for each of the groups. So on our left, we have our upregulated genes, so the more dark blue, purple of the circle is going to be, the more strong that statistical significance is going to be. Whereas on our right, with the downregulated group, the yellow indicates the more statistically significant a gene ontology group will be. And the proximity of these groupings together indicates their similarities between one another. And what we found in these figures particularly is that biological process, metabolic process, and cellular process are found in both our up and downregulated groups. But when we see an upregulated, biological process and metabolic process are much more closely related or close together compared to our cellular process and our metabolic process proximity in our downregulated figure. This just highlights that our gene ontology groups are generalized groups that contain numerous genes within them. Now, looking at our gene ontology figure for the attached versus detached trophozoites, looking at the upregulated, so these are genes that are going to be more expressed in attached trophozoites than in detached. We see a lot more metabolic associated genes, polyol catabolic process, energy reserve metabolic process, to name a few. But looking at our downregulated genes found more so in detached rather than attached trophozoites, we see that the detached trophozoites, as they're preparing for reproduction, are having a greater expression of genes associated with meiosis and DNA replication, DNA topological change, dephosphorylation, and intracellular signal transduction. Again, here we see a gene ontology pathway figure highlighting that relationship in a more visual form. And lastly, we are looking at syzygy versus detached trophozoites. And these tree maps look a little different than the ones from the previous slides. As you can see in our upregulated tree map, there's not as many genes highlighted within this figure. If you remember from the differential expression analysis from a lot of slides ago, our syzygy and detached trophozoites had the most similar genes compared to the other two comparison groups. And this just highlights how our detached trophozoites are moving into that syzygy stage, but they're just waiting for that extra gargant to pair up with. But there are two key groupings here: cell redox homeostasis, which helps with the maintenance of the cells, and amino sugar metabolic process, which helps with protein regulation. And lastly, when looking at our downregulated tree map, we see that, once again, our detached trophozoites are preparing for that reproductive stage, so they have a greater expression for these preparation genes: DNA replication initiation, maintenance and location, cellular component organization, protein-containing complex organization, and cell cycle. Once again, here we see that same subcategories from the previous slide in a more visually appealing form. Now, to conclude, I looked at three main comparison groups: the syzygy versus attached, attached versus detached, and detached versus syzygy. Through these comparison groups, I was able to see the relationships between them, but also gain a broader description for each stage. For our attached trophozoites, the feeding stage, I saw a lot of genes associated with metabolism. For our detached trophozoites, those in the embryonic stage, preparing for reproduction, I saw a lot of those preparation genes associated with DNA replication. And lastly, for syzygy, the reproductive genes, I saw a lot more genes associated with homeostasis and maintenance. So before I finish, I would like to take a moment to thank my three advisors, Dr. Gould, Dr. Hauser, and Dr. Rowe. As stated, I've been working on this project for the past 2 years, with Dr. Gould by my side for the majority of it. And after his passing, Dr. Rowe so graciously stepped up to the role and has been an amazing advisor this past year. I would also like to thank the McNair Scholars Program, the I4 Institute, and St. Edward's Natural Science Department for their funding of this research, and all my past school lab members who have helped me along the way. This is the slide that was supposed to be there. Does anyone have any questions?
questions first, and then we'll ask Dr. Rowe to come up. But are there questions for Mia?
I like how the decor of the room, even the lamp covers the room. Do all cockroaches have that parasite? And if so, how do they get it?
So all cockroaches can contain a Gregarine parasite. This Gregarine parasite in particular only infects Madagascar-hissing cockroaches. I'm not sure on the reasoning for that, but we used this model of host and this specific species of parasite because of that one-to-one relationship. So any Gregarine that we were isolating from this host, we ensured was the same species.
So I know parasites are weird, but I'm just trying to wrap my head around it. Since it's used in the reproductive stage, so they basically going through meiosis? If that's what's happening then?
Yeah.
Okay, so then, because that's the sexual reproduction, yeah, great.
I have a question about the figures. Rotating circles. Is that kind of the round figure or the round shape of the figures? Does that represent the life cycle, or is it just kind of how it works out?
It's just how the variation worked out. So I can go back to kind of highlight the X and Y axis are just somatic space, and that's just a generalized form to kind of highlight the variation of them. But it's not based off of any values per se.
Was there anything you found of surprising based on your knowledge of parasites?
I was really interested in the amount of metabolic genes that I found in the attached trophozoites. I'll just go back to this figure. But yeah, we knew that the attached trophozoites had that direct relationship with the host. And I was really interested to see how the attached trophozoites versus detached trophozoites varied in their gene expression, because they're the same life cycle stage in the typical looking at our Gregarine parasites, but they did have very different gene expressions. So it's interesting to see them categorized in the same way.
So thinking of prevention, if you look at attached versus detached, do you see anything turning off in the detached that would maybe indicate how they integrated with the host cell?
I'm not sure. That would, that's an interesting point. I would want to look more further into it. Me and Dr. Rowe made these figures last week. That's why I was looking at my notes.
It just might not be the information in the host situation.
Yeah, that was absolutely incredible. Amazing presentation. It sounds like really incredible research, and I'd love to have Dr. Rowe come up and give you this award and soul to represent this amazing project, but also everything you've contributed to the honors program while you've been here. It's been our honor, and I hope you'll wear these with pride at graduation.
I just have to say, so last fall Mia came to me and asked me if I could be her mentor. And I went and I spoke with the other faculty because this was my first time mentoring a student. And it was told to me that Dr. Gold would often refer to her as one of the most intelligent, wonderful students he's ever worked with. And I talked to multiple faculty members that could say nothing but wonderful things about Mia. And I can honestly say after working with her this semester that they undersold her abilities. And I can say without a doubt when she gets to Harvard this fall, she will succeed with flying colors.